In protein synthesis, there are two major processes. One of those processes is transcription. This occurs when DNA is transcribed into RNA. The resulting mRNA will be complementary to DNA. Transcription also takes place in the nucleus. After transcription is the process of translation, which takes place in the ribosome. This occurs when RNA is translated into a protein or polypeptide. Some of the proteins that we know of include methionine and serine. So the first step of protein synthesis is transcription. This is when we take a gene and transcribe it into RNA or mRNA. Afterwards, translation takes place and mRNA is translated into a protein. Let's look at the major differences between RNA and DNA. mRNA, or messenger RNA, is single-stranded, shorter than DNA, and has uracil instead of thymine. It's necessary to copy DNA to RNA so they can leave the nucleus. On the other hand, DNA is composed of a double-stranded double helix. It is also longer than RNA and it contains thymine. Protein structures include primary protein structures, secondary, tertiary, and quaternary. There are four major components of protein structure. For primary protein structure, there is a sequence of a chain of amino acids. Secondary protein structure consists of beta-pleated sheets as well as alpha helices. Secondary protein structure requires folding the polypeptide chain into sheets or helices. Tertiary protein structure is consisted of a beta-pleated sheet as well as alpha helices. There is also three-dimensional folding pattern due to side chain interactions. In the case of quaternary protein structure, the protein consists of more than one amino acid chain. Base pairing in DNA is also known as Watson-Crick base pairing. Adenine always pairs with thymine and cytosine always pairs with guanine. Adenine and guanine are both purines and cytosine and thymine are both pyrimidines. We know that there are two hydrogen bonds between A and T between adenine and thymine, and there are three hydrogen bonds between cytosine and guanine. This also contributes to the double helix structure of DNA. DNA has a sugar phosphate backbone as well.